from technician to technician and from plant to plant. The majority of food companies use Petrifilm plates in both their large and small plants to optimize their labor productivity and increase their profitability. Efficiency is paramount in today's manufacturing facility, and since labor cost is the largest expense related to microbial testing, companies worldwide are recognizing the dramatic difference 3M Petrifilm plates can make. With a documented average labor savings of 45% versus conventional auger plate use, it's easy to see why 3M Petrifilm plates are the logical choice as you strive to manage your lab resources more effectively. Petrifilm plates help maximize laboratory productivity and efficiency, while delivering the consistent, accurate counts that help address regulatory issues, especially since Petrifilm plates have AOAC official methods approval. There are 4 basic steps to microbial testing: sample preparation, sample plating, incubation, and interpretation. For liquid samples, 1 milliliter of the sample can be plated directly onto Petrifilm plates. Some products may require a dilution. If you need to dilute your sample, do so per your usual methods. You may want to refer to the 3M Dairy and Juice application sheet or our Guide to Dilution Preparation for recommended dilutions. For solid and semi-solid samples, also refer to this application sheet for dilution preparation and techniques. When the samples are prepared and ready to be plated, choose from a full line of 3M Petrifilm plates: Aerobic, coliform, E. coli coliform, Enterobacteriaceae, high sensitivity coliform, rapid coliform, rapid Staph aureus, Staph express, and yeast and mold. With each type of Petrifilm plate, an interpretation guide with reminders for use is provided to guide you through storage, sample preparation, pH adjustment, plating, incubation, and interpretation of your test results. There are 3 methods of plating depending on which Petrifilm plate you use. For the Petrifilm plates that feature a foam dam to contain the inoculum, such as the coliform, E. coli, rapid Staph aureus, rapid coliform, Enterobacteriaceae, and Staph express count plates, here is the recommended procedure. Label the plate and place it on a flat surface. Then attach a tip to your pipetter. We're demonstrating with 3M's 1 milliliter electronic pipetter and tips, a pre-programmed instrument for diluting and pipetting onto 3M Petrifilm plates. More information on this instrument will be covered in the subsequent video module. Draw 1 milliliter of sample. Then lift the top film and hold the pipetter perpendicular to the Petrifilm plate. Pipette the sample onto the center of the bottom film. Carefully roll the top film down to avoid entrapping air bubbles. Pick up the appropriate spreader with the flat side down and place it on the top film over the inoculum. Then gently apply pressure on the spreader to distribute the inoculum over the entire circular area. Do not twist or slide the spreader. Lift the spreader and allow 1 minute for the gel to form. Since the Petrifilm yeast and mold and aerobic count plates do not feature a foam dam, the plating procedure is a little different. Place the labeled plate on a flat surface. Then draw 1 milliliter of the diluted sample into the pipetter. Lift the top film and hold the pipetter perpendicular to the Petrifilm plate. Pipette the sample onto the center of the bottom film. For best results, release the top film and simply allow it to drop. Pick up the appropriate spreader with the ridge side down and place it on the top film, centered over the inoculum. Apply pressure on the spreader to distribute the inoculum over a circular area. Do not twist or slide the spreader. Lift the spreader and allow 1 minute for the gel to form. The third method of plating applies only to 5 milliliter Petrifilm high sensitivity plates. Place the labeled plate on a flat surface. Using 3M's 5 milliliter electronic pipetter and tips, draw 5 milliliters of sample. Lift the top film and pipette the sample onto the center of the bottom film. Gently roll the top film down onto the sample to prevent pushing the sample off the film and to avoid entrapping air bubbles. Place the high-sensitivity spreader on the top film over the inoculum. Then, distribute the sample with downward pressure on the handle of the spreader. Do not twist or slide the spreader. Lift the spreader and allow 2 to 5 minutes for the gel to form. Before we demonstrate the incubation process, a word is needed about storing and disposing of 3M Petrifilm plates. Refrigerate or freeze unopened packages and use them before the expiration date on the package. Seal opened packages by folding the end over and taping it shut. Do not refrigerate the package once it's open. Simply keep it in a cool, dry place and use it within 1 month after it's been opened. These instructions should be followed for all Petrifilm plate products with the exception of the Staff Express discs. Open pouches of Staff Express discs must be resealed and can be stored frozen for up to 6 months. After use, Petrifilm plates may contain viable bacteria. Follow current industry standards for disposal. Now let's move on to talk about incubation. Incubate Petrifilm plates with the clear film side up in stacks of up to 20. For rapid Staph aureus and high-sensitivity plates, limit stacks to 10 plates or less. The temperature of the incubator and the amount of incubation time needed will vary depending on which plates you're using. Refer to the product's package insert for specific instructions. The 3M Petrifilm Rapid Staph aureus count plate method consists of 2 parts. The plate itself, which contains modified Baird-Parker nutrients with a gelling agent, and the thermostable nuclease, or TNase, reactive disc. An extra step is needed in the incubation process. After the plate has been incubated for 24 hours at 35 degrees Celsius, further incubate the plate at 62 degrees Celsius for 1 hour. This will allow you to differentiate Staph aureus having thermostable nuclease from other bacteria. Lift the top film and place the blue TNase disc in the well. Replace the top film. Now, re-incubate the plate at 35 degrees Celsius for 1 to 3 hours. The 3M Petrifilm Staff Express count plate method also consists of 2 parts. The plate itself, which contains chromogenic modified Baird-Parker nutrients and a gelling agent, and the Staff Express disc to facilitate differentiation of Staph aureus from other suspect colonies when background microflora are present. The Staff Express disc may or may not be needed. If no colonies or only red-violet colonies are present after incubation at 35 degrees Celsius for 24 hours, the test is complete and the disc is not needed. If colony colors other than red-violet are present after the 24-hour incubation, lift the top film and place the blue Staff Express disc in the well. Replace the top film and then re-incubate the plate at 35 degrees Celsius for 1 to 3 hours. The final step in microbial testing is interpretation. Good lighting is necessary for accurate counting. An illuminated magnifier and a hand tally may help for counting bacterial colonies. As mentioned earlier, 3M Microbiology provides an interpretation guide for each type of Petrifilm plate. We'll briefly review tips for interpreting each type of plate. The dye in 3M Petrifilm coliform count plates colors all colonies red, and the top film traps gas produced by coliforms. Coliforms produce red colonies associated with gas bubbles. Non-coliform colonies are red, but are not associated with gas bubbles. Do not count colonies that appear on the foam dam because they're removed from the selective influence of the medium. In addition to the components of the coliform count plate, the 3M Petrifilm E. coli coliform count plate contains a glucuronidase indicator dye, which forms a blue precipitate around E. coli colonies. Most E. coli also produce gas, which is trapped by the top film. Count the blue colonies associated with gas bubbles as E. coli. The total coliform count can also be determined by counting all red and blue colonies associated with gas bubbles. For 3M Petrifilm Rapid Staph aureus count plates, detection of TNase is a confirmatory method for Staph aureus. An indicator in the Petrifilm TNase reactive disk causes colonies to appear red or blue. A second indicator causes pink TNase zones to appear, which confirms Staph aureus. Count even small pink zones. Both of these examples have about the same count, though the pink thermostable nuclease zones vary greatly in size. The Staph Express count plates contain chromogenic media which is selective and differential for Staph aureus. The indicators in the Staph Express plate cause Staph aureus to appear as distinctive red-violet colonies. If no colonies are present after 24-hour incubation at 35 degrees Celsius, the test is complete. If only red-violet colonies are present after the 24-hour incubation, count those colonies as Staph aureus and the test is complete. If colony colors other than red-violet appear, as in this example, use a Petrifilm Staph Express disk to differentiate Staph aureus from other suspect colonies. An indicator in the Staph Express disk causes Staph aureus colonies to develop a pink zone surrounding the colony. After re-incubating the plate and disk for up to 3 hours at 35 degrees Celsius, count colonies with pink zones as Staph aureus. 3M Petrifilm rapid coliform count plates contain a pH indicator, which detects acid produced by coliforms, and an indicator dye which colors all colonies red. Also, the top film of the plate traps gas produced by the coliforms. Early coliform results may begin to appear within 6 hours of incubation and appear as discrete yellow acid zones with or without colonies. Final coliform results appear as colonies associated with gas bubbles and may begin to appear within 8 hours of incubation. At any time within the 24-hour incubation period, you see a colony associated with gas. It's a confirmed coliform. Continue to incubate plates after each reading to detect confirmed coliform growth. A total coliform count is determined at 24 hours. 3M Petrifilm Enterobacteriaceae count plates have 3 indicators to assist you in interpreting the plate. An indicator dye in the plate colors all colonies red. The top film traps gas produced by some bacteria, and the pH indicator detects acid production. Acid-producing bacteria are seen as red colonies surrounded by yellow acid zones. Enterobacteriaceae have the following characteristics: colonies associated with gas bubbles but no acid zones, colonies with yellow acid zones but no gas bubbles, or colonies producing both gas bubbles and yellow acid zones. When bacterial colonies are present after incubating 3M Petrifilm aerobic count plates, an indicator dye in the plate colors the colonies red. All colonies should be counted, regardless of size or color intensity. Petrifilm aerobic count plates can also be used to determine a lactic acid bacteria count. Anaerobic incubation using MRS broth gives you the ability to differentiate between gas-producing and non-gas-producing bacteria on the same plate. Refer to the procedure sheets on lactic acid bacteria for further instructions. 3M Petrifilm yeast and mold count plates contain an indicator dye to provide contrast and facilitate counting. Yeast typically form small colonies with defined edges. They also may appear three-dimensional and do not have center foci. Molds are often larger, variably colored, flat colonies with diffused edges and center foci. With 3M Petrifilm high-sensitivity coliform count plates, an indicator dye in the plate colors all colonies red, and the top film traps gas produced by coliforms. The preferable counting range varies for each type of Petrifilm plate. Sometimes it may be necessary to estimate the count. To do this, first determine the average number of colonies in a square. To calculate the average, count 2 or more representative squares, then divide that count by the number of squares used. Multiply that number by the size of the inoculated area on the plate to obtain the total estimated count per plate. The size of the inoculated area may be 20, 30, or 60 square centimeters. See the product package insert or interpretation guide for more information. In addition